Hey, hi. So we are done with the basic introduction for this particular chapter, and the very first thing that we are starting 6.1 and the DNA structure. Okay, so we are going to deal into the structure. Full form of the DNA, as we know, is deoxyribonucleic acid. Okay, nothing fancy about it. Let's try to find out that what is it composed of. So the thing that it is composed of is number one, sugar. Okay, in simplified version. Number two, that it is composed of nitrogenous bases, and the number three, that it is composed of phosphate. Biology is nothing but the chemistry onto the basic level. So this is phosphate. Now, what kind of sugar? There are so many different kinds of sugar. Glucose is a 6-carbon sugar and other kinds of sugar. What particular kind of sugar are we talking about right here? So we are basically talking about a 5-carbon sugar. Okay, so we are talking about 5-carbon sugar, and 5-carbon sugar specifically that is being utilized into DNA and RNA is that if it is ribose, okay, if it is ribose, which is a 5-carbon sugar, it makes the RNA. And if it is deoxyribose, then it becomes the for the DNA. So the basic structure, it does look like this. Okay, so this is a 5-carbon sugar. So 1, 2, 3, 4 CH2OH. So this is the basic structure of a 5-carbon sugar. And let me give the carbon number 1, 2, 3, 4, 5, and this is just a convention that we name it by dash 1 dash 2 dash 3 dash 4 dash 5 dash. So this is how we are going to refer that carbon onto the DNA, and these dash actually represent that we are talking about specifically the carbon that are present into the sugar only when we are talking about the DNA. Okay, so that we do not get confused into some other carbon that is present onto, let's say, onto the nitrogenous basis, but we are talking about specifically onto the sugar carbon. So here we have got a 5-carbon sugar. If you have got OH right here, then it is known as the RNA, and if it does not have OH, it only have what H, then it is known as the DNA. Okay, no, not known as, but this is the what you could say that it is the integral part, and in which particular form it is present is defining whether it will be a RNA or whether it will be a DNA. So if O is there, then it will be RNA, and if O is not there, which the name itself of the DNA is suggesting deoxyribose, okay, so there is the name itself is coming, so this will become the deoxyribose sugar. Now when sugar and a nitrogenous bases, okay, let us first discuss the nitrogenous base. Okay, so nitrogenous bases can be divided again depending upon the number of rings they have. So if they either they can have 2 rings into their structure, or they can have 1 ring. So if it 2 rings, then it is known as purine, and if it is 1, then it is known as the pyrimidine. Okay, the purine itself is can be divided into 2, which is adenine, which is also denoted as A, and the guanine, which is denoted as G. Pyrimidines can be divided into 3. 1, 2, cytosine denoted as C, thiamine, which is denoted as T, and uracil, which is denoted as U. Okay, so cytosine is present in both DNA as well as in RNA. Thiamine is only present in DNA, and uracil is only present in RNA. This is the basic general classification of the DNA onto the chemical level. So we are not going into the structure first. So this is what they are composed of. Let us try to find out that what is going to happen if your sugar and nitrogenous base bonds with each other. Okay, so if sugar and nitrogenous bases do bond with each other, then what is going to happen? So let us say that ribose is not going to change. So we have got this sugar, which is deoxyribose sugar, as it is sugar, and this is going to make a bond with nitrogenous bases. Okay, so this will be the nitrogenous base. This nitrogenous base can be what? Either can be adenine, it can be guanine, it can be cytosine, it can be thiamine. So this whole structure, so nitrogenous bases along with the sugar molecule is known as your so we have to give their respective names. So their name would be, for adenine it will become adenosine, and if it is bonded with guanine then it will become the guanosine, and if it is with the cytosine it will become cytidine, and if it is thymine then it becomes thymidine, and one which is remaining, if it is with uracil then it becomes uridine. Okay, and the bond by which it is being bonded is known as N-glycosidic linkage. N-glycosidic link. This whole structure is known as a nucleoside, and when the sugar is also get attached, okay, also get the phosphate, let us say right here, this is the phosphate, then it becomes the totally nucleotide. What does it become? Nucleotide. And nucleotide is the monomeric unit for the DNA, monomer for DNA or RNA, you can say, depending upon that what particular group, functional group that you have. If you've got uracil, then it is RNA. If it is thymine, that it is DNA. Okay, what other differences we are going to see along with it. Okay, so this bond will be known as phosphodiester bond. Phosphodiester bond. So if the bond between sugar and nitrogenous bases is known as the N-glycosidic linkage, the bond between the sugar and the phosphate is known as the phosphodiester linkage. Okay, let us try to validate all this information that we have gathered with the NCERT. Okay, so let us try to validate all the information that we have gathered with the NCERT. Okay, so I'm going to read it. Let us recapitulate the chemical structure of the polynucleotide. So we have discussed the detailed structure of the DNA and RNA. So DNA and RNA, which is composed of what? The nucleotide has three components. Number one, nitrogenous bases. So there we have got nitrogenous bases, and the pentose sugar. There is the pentose sugar, and if it is 5 carbon, it is a 5 carbon sugar. If it is ribose, then it is a component of RNA. If it is deoxyribose, this is a component of DNA. Okay, and the phosphate group. Okay, so the phosphate group is right here. There are two types of nitrogenous bases. Okay, two types, fine. There it is. So purines, and which can again divide it into adenine and guanine. So adenine and guanine. Pyrimidines into cytosine, uracil and thymine, out of which cytosine is present in both uracil in RNA and thymine in DNA. The same line is right here. And after that we have got N-glycosidic linkage to form a nucleoside. So when sugar bonds with a nitrogenous base through the N-glycosidic linkage, this becomes the nucleoside. So as you can see right here, we have got the nucleoside. If it is a adenosine and the vice versa, all those things, and onto the 3-OH group and the 5-OH group, we have got phosphate bonded to it with the phosphodiester bond, but that we are going to see into detail into when we are going to deal with the structure of the DNA. Right now we are dealing with the component, that what is it being composed of. So a phosphate is bonded with sugar with the phosphodiester bond. At what position? 3- and 5- we are going to see later. Okay, so two nucleotides are linked through the 3-5-OH. So this polarity, so-called polarity. So please remember this: 1-, 2-, 3-, 4-, 5-. So one phosphate is bonded right here onto this 5- group, 5-OH, 5-CH2OH, and one is bonded right here onto the 3-OH group. And there comes the polarity. We'll discuss it. So let us move on to the next thing. Okay, let us try to read this as well. So we have already discussed that in RNA every nucleotide residue has got an additional OH group. So we have discussed it, and the polarity we are about to discuss. But just before we discuss the polarity, this is the basic history of the structure of the DNA. The chemical nature of the DNA was known much earlier, but the structure was not known. So these were the scientists: Rosalind Franklin, Maurice Wilkin, James Watson, Francis Crick. James Watson, Francis Crick and Maurice Wilkin got the Nobel Prize for the double helical structure of the DNA, but not the Rosalind Franklin, and that happened in year 1953. But the Rosalind Franklin was the key scientist for the discovery of the double helix DNA. So she was a X-ray crystallographer. So what did she do? She made the crystal of the DNA, and she looked it, and she found out the X-ray crystallogram of those crystals. And those crystallograms were read and being deduced by James Watson, Crick, and all these peoples. And those two people that we actually know from 1953 and the structure of the DNA, the Watson and Crick double helical model of the DNA. Okay, so this was the basic history of the DNA, history of the structure. Now let us move on to the polarity and stuff. Okay, so let us say this was my ribose sugar. This will be my 1 dash, 2 dash, 3 dash, 4 dash. So this is my, and this is CH2OH. This will be my fifth carbon. Onto 2 dash carbon, I will have only H into DNA. I'll have onto 3 dash, I will have OH. This OH is going to make bond with phosphate. Okay, and this and also it is going to make bond with the next nucleotide. Okay, so this is how it is going to make the bond. So this is the next sugar, and this is my CH2OH. So this is going to make bond with this 5 dash carbon of the next sugar. And right here again I'll have a 3 dash OH group. This is going to again make a bond with phosphate, and this phosphate again going to make a bond with OH of the fifth carbon. Okay, and this is how it is going to continue to make a polymer. Okay, so this is hence comes the polarity from 5 dash to 3 dash, because you have got 5 dash OH free right here, and you have got a 3 dash OH group free right here. So this will be a 5 dash polarity, and this will be a 3 dash polarity. Okay, this is how this is one strand of the DNA. On to the sugar itself, we have got our nitrogenous base attached onto it, isn't it? So this is my nitrogenous base, this is also my nitrogenous base, this is also my nitrogenous base. Nitrogenous base is making bond with other nitrogenous base. So on to right here I'll have another nitrogenous base, another nitrogenous base, next nitrogenous base. The important point is that just as it was going 5 dash direction to the 3 dash direction, here it goes into opposite direction. So right, I will have 5 dash OH group free onto the lower end and 3 dash free group onto the upper end. Let us try to make it. So why is it happening? Because of the inverted nature of the sugar. So right now I have got my sugar which is somewhat looking like this. Okay, so I have got this is my first carbon, right? This is my first carbon, and my 3 dash OH is free into this particular situation. Carbon number 1, carbon number 2, carbon number 3. This OH is free. This carbon CH2OH is going to make a bond with phosphate, and this phosphate again going to make a bond with the OH. Okay, again 3 dash OH of the next carbon. So this will be my sugar. This will be my sugar. This is my attachment of nitrogenous base. This will be my OH of 3 dash OH, and this is how this is going to continue and get along. So now you know that what is 5 dash and 3 dash polarity. It means the DNA strand has got 5 dash OH group free and 3 dash OH group free. Okay, so this is the polarity. Either it can be 5 dash to 3 dash, or it can be 3 dash to 5 dash. Okay, so this was the structural aspect of the DNA. After that, there was a scientist named by Erwin Chargaff, and what did he find? That he said that the amount of adenine inside a cell is somewhat, not exactly, it's somewhat equal to thymine, and the cytosine is somewhat equal to guanine. Okay, somewhat equal, okay, not exactly equal, somewhat equal. So what did he say? That if he divided by A by T, which is equal to C by G. Why? Because they both are equal, so it will also come 1. This is also come 1. So A by T is equal to C by G, which is equal to 1. According to him, this is known as the Chargaff's rule. And by which what we can say that the number of A is equal to number of T. It means that A must be pairing with the T, isn't it? So A must be pairing with the T with 2 double bonds. This is one thing. The next thing would be the cytosine must be pairing with the guanine, right? So but it bonds with it with 3 bonds. So this is the next thing. So if it is A, then it is going to pair with T with double bonds. And if it is C and this is G, then it is going to pair with it with each other with 3 hydrogen bond. Okay, so now here comes the salient features of the double helical DNA. Or so one, let me take up the highlighter. So it is made up of 2 nucleotide chain. One is 5' to 3', one 3' to 5', where the backbone is consisting of sugar and phosphate. We have seen that sugar and phosphate making the backbone, and the bases are projected inside. Their bases are projected inside so that they can make the hydrogen bonding. The two chain have anti-parallel polarity, 5' to 3' and 3' to 5'. We are done with this thing as well. The next para is the bases of 2 strands are paired through the hydrogen bond. So these bonds are known as the hydrogen bond, and forming of these base pairs, adenine forms with 2 hydrogen bond with thymine. So A bonds with T with 2 bonds, and cytosine makes 3 bond with the guanine. So forms of the opposite and the vice versa. Similarly, guanine is bonded with cytosine with 3 hydrogen bonds. We are done with that. As a result, always a purine comes with opposite to pyrimidine. So can you see specifically that purines comes opposite to pyrimidine? Adenine and guanine are purines, and thymine and cytosine are pyrimidine. Uniform distance between the 2 strands of the this actually makes the distance between. So if there is space of 3 rings, okay, there is a space of 3 rings. If let's say I have added a purine which is a 2 ring, only 1 ring space is left, so pyrimidine has to come. If I have added pyrimidine, then the space for 2 ring are left, and then I'll add a purine. So this makes a constant distance between 2 backbones. The 2 chains are coiled in the right-handed fashion. Okay, they are coiled into a right-handed fashion that you are supposed to see the 3D structure of. So this is the double helical structure of the DNA. As you can see that if this is the backbone, this is the direction in which you are going. So if you place your, let me try to make it. So if I place my thumb upside down like this is my thumb, then it is going, my fingers are curling inside, right, isn't it? So my fingers are curling inside. So this is known as the right-handed helix of the DNA. Okay, so this is the right-handed helix. So if my thumb is showing the direction in which it is going, and that fingers will show that it is going into this particular direction. Okay, so this will be known as the right-handed helix. Okay, why we have got right-handed helix? Because there has to be a left-handed helix as well. That is why we have 2 different kinds of helix in our DNA. One is the left-hand helix and the one is the right-hand helix. This one is specifically right-hand one. The next thing, the plane of one base pair is stacked over another in double helix. So as you can see, this is one base pair is which is stacking over each other, and this in addition to hydrogen bond conferred to the stability of the helical structure. This also stabilizes the DNA. This is just like a staircases thing, that you have got a staircase, you have got the backbone, as well as these will be the steps of the staircase. So can we compare? So we're done with the one particular topic. This is known as the central dogma of the biology. Okay, the one thing that we have discussed in our first video, that we have to discuss the functional aspect. Okay, the functional aspect will be first that it can replicate itself to propagate the information from one generation to next generation. Next, that DNA is an information for what? To make protein. And there is a in between thing which is known as the mRNA. What is mRNA? If inside the nucleus you have got this DNA, this is the DNA. Please consider this as the blueprint of your to make the protein. This is the blueprint. But what if you lose the blueprint? You don't have anything to refer to. So what happens, that a photocopy can be taken, and that photocopy is known as the mRNA, and mRNA can be sent out. Even this copy is being lost by the cell, then you have got the blueprint. You can make the protein again. So mRNA can act as the photocopy, and DNA is acting as your blueprint. So this is known as the central dogma of biology, in which DNA is being utilized to make the mRNA. Okay, it is not converted. This is important. And mRNA is being utilized to make the protein. Okay, so we are done with 6.1.1. So we'll move into next video with 6.1.2.